Bioprinting is the three-dimensional printing of biological tissue and organs through the layering of living cells. It starts with the fundamental structure of the specific organ being uploaded into the bioprinter so it can create the correct organ. The bioprinter then starts to deposit thin layers of cells onto the mold using a bioprint head that moves left to right, forwards to backwards, or up and down. A special ink is used to dispense the cells, which is called bioink. Bioink is a special material made out of real living cells that behaves like a liquid. A substance called hydrogel is dispensed at the same time that helps support and protect the organs being created, and when it is done, it dissolves. As you can see, a layer of hydrogel is placed down to support the cells. Then the bioprinter starts to place down the cells onto the hydrogel. A second layer of hydrogel is placed down to support the organ, and then more cells are added. This process is repeated until the organ is done.
A team of Canadian researchers create artificial blood vessels by wrapping sheets of cultured human cells into tubes. There's no printing involved, but the achievement is an important step in 3D tissue engineering. Thomas Bolin modifies an inkjet printer to deposit biological material such as proteins and bacteria. He continues to develop the technology and earns a broad patent for inkjet printing of biomaterials in 2006. An international team of researchers prints tissue scaffolds by extrusion, a process that pushes gels through nozzles to build 3D structures layer by layer.
Organovo and Inventec create the first commercial bioprinter, the NovoGen MMX.
Wake Forest researchers bioprint skin directly onto injured mice to help heal burns. Princeton University researchers led by Michael C. McAlpine bioprint a working ear.
Organovo releases 3D printed liver tissue to aid in drug development. We're trying to make something that is superior to animal trials, says Keith Murphy, Organovo's CEO. Every pill that is used today needs to be tested first. These tests are usually done on mice and stuff, but say a heart pill is tested on a mouse. The mouse's heart and mine is not the same. Their build is different. So if we could bioprint a heart and test a drug on that, we could get more accurate results and we could save animals from being tested upon.
The pros to bioprinting are faster and more precise than traditional methods of building organs by hand, less likely to be prone to human error, less laborious for scientists, less likely to be rejected because the organ is made from the cells of the patient, decreased waiting times for organ donors. Drugs can be tested on the bioprinted organs instead of animals, so it's more humane. Finished products are independent of biomaterial or scaffolding absent in native tissues. Effects of disease states or drugs may be more accurately observed without the need for human subjects. Well-organized, diverse cell types allow enhancement of tissue-specific functions.
The cons of bioprinting are questions of viability if a printed object fails, disputed ownership of the codes and implants produced, various ethical concerns, pricing available only to the wealthy, consumption of large amounts of energy, emission of unhealthy particles into the air, difficult in maintaining cell environment, resulting in the death of many of the cells created. As you can see on your screen right now, this is what happens when you get or experience an asthma attack. When you are exposed to a certain allergen, such as a tree, grass, or weed pollen, this muscle constricts your airway, so it makes it more difficult to breathe. The scientists have printed out this muscle and exposed it to a certain allergen. The result is that it contracts. This just proves that we can test drugs on it and get accurate results.
There are much more details about bioprinting that are out there and much more development that is required to make this an everyday luxury. Thank you for watching our video about bioprinting of organs and tissues.